tuning in. We have 26 participants now, so I think we're certainly getting there. So maybe we can. I'll just say a few words. It's extremely exciting to observe what's going on at Northwestern with synthetic biology. In the field of synthetic biology, this phenomenal center that Milan started, and which has been continued, and I'm sorry, Milan, making it better and better by people like Jewett, and thanks to work of our exceptional faculty like Josh. This is such an incredible opportunity for Northwestern to be really world leader in synthetic biology, and especially in cancer, and that's the focus of today's meeting. One of the ideas is behind what you're going to hear today is to develop new collaborations, bring in experts in clinical medicine, diagnostics, therapeutics, translational medicine, is particularly related to cancer, and link those with our, like I said, world leaders in synthetic biology. NIH is certainly interested in fostering synthetic biology and cancer. They view this as the next big quantum leap in cancer research and therapeutics in particular, just like nanotechnology emerged as a significant field within the significant technological driver in cancer research a couple of decades ago. There's a recognition at the level of NCI that synthetic biology may fill this role and is beginning to fill this role. So what I hope we can get out of this meeting is an exciting conversation about where synthetic biology will stand in cancer research. I would like everybody to ask as many questions as you can, and please feel free to follow up with any of our fantastic speakers today, offline or during this meeting. Please use chat, and hopefully some connections will be built as well. So a few words about the logistics. So we have 3 talks. This is a very fast-paced session, 10 minutes each. Milan will present first. Followed by Mike Jewett, and followed by Josh Leonard. 10 minutes each. After each 10-minute presentation, we'll have 5 minutes for Q&A, specifically related to that talk, and after that, we'll have another 10 minutes of a panel discussion where you will have chance. Every attendee will have a chance to ask questions to any of the 3 speakers. So what I suggest we do when you have questions, just type it into everybody, or to me personally, if you prefer, within the chat box, and then I will call on you to ask the question after the talk is over. And with that, without further ado, I would like let Milan give his 10-minute talk. His talk is entitled "Megamolecule Mimics for Therapeutic Antibodies."
Okay, great. Thank you, Vadim. And I know these are short talks, so we won't get into a lot of details, really just give you a sense. But I think, the general excitement at Northwestern over having such a strong synthetic biology group, one of the absolute strongest in the country, and where a lot of the applications in synthetic biology are actually in cancer. There are many others, but one key one is cancer. So seeing how to bring this together and seeing whether this can develop into one of the new strengths, it's a key part of a renewal of the CCSG, I think could be one of the productive outcomes here. So I want to talk about megamolecules. These are molecules we've named. They're extremely large and they're extremely simple to put together. So let me at a high level say that the building blocks here, the Lego pieces, are fusion proteins and simple organic linkers, synthetic linkers. And by going through steps where you join pieces together, you can build structures that are really long. They're protein-based, but they have these linkers too. And so they're extremely modular and have the ability to create scaffolds, which are very large, but then are decorated and functionalized with a whole variety of groups, whether they're proteins and fabs, whether they're enzymes, whether they're nanoparticles, fluorescent labels, catalysts, whatever. Now, the key thing here is how do we put these together? Well, we use reactions of enzymes and covalent inhibitors or irreversible inhibitors. And so this is one system. Cutinase is covalently inhibited by these phosphonate esters. And there are three reasons this is really exciting. One is that the ligands are actually not that reactive. They don't react with other things. They only become reactive when they're in the active site of their target enzyme and the catalytic machinery activates for reaction. So you only get reaction where you want it. The second is that they're extremely fast because even if the pieces are present at low concentration, they come together through a binding, like a KM. And so the binding brings them together so that they can react quickly, and our reactions typically take minutes, tens of minutes. And the third is that they're very specific. We don't see reactions of these groups outside the active site residue that participates in the reaction. So we have a lot of these pairs of reactions: cutinase, halo tag with its alkyl chloride, snap tag with its benzyl purines, and many others. And the way you build structures, if you take that fusion protein on the left, it turns out to be one halo tag and one cutinase. But if you treat it with a linker, the blue covalent inhibitor reacts with the blue domain. Treat it with another fusion protein, gold reacts with yellow. Another linker plugs in, fusion protein plugs in, and you're building these structures, 20, 30-minute reactions a piece, high yields, really simple. You can take that guy and dimerize these two blue domains with the linker, which is symmetrical, blue-blue. Now you take this structure and load another linker on each end, snap on another arm, and you create a structure which has a molecular weight of about 650,000 Dalton, far beyond what you can make with recombinant expression, and dimensions in excess of 100 nanometers, but with perfect control over structure. We know where every bond is and every atom is. And you can, you almost think about this as shipbuilding in a bottle. Here's a linker which is 3-armed. Each of the arms has a chemistry that will only react with a given enzyme. So if you take that linker and you add it in a reaction with the 3 enzymes, each one will snap in place only where it's supposed to go. So the ability to create these structures efficiently with, again, perfect control has benefits in a lot of areas. Our primary interest is in antibody mimics for immunotherapy. And so let me just remind everybody what they know, which is on the left is the consensus structure for the IgG motif, 2 fabs. On the right is a megamolecule mimic of an IgG, where we bring 2 fabs together by creating a fusion protein of the fab with cutinase, another fusion protein of a fab with snap tag, and using a linker that plugs those in. And so we did this, and we did it for anti-HER2. And so we were building a trastuzumab analog out of our megamolecules. The fab domain was exactly the same as the fab that's present in trastuzumab, the approved drug. And so the point is we can make the fusions, we can make the linker, we can snap things together, we can purify that structure. We do a lot of characterization. Here are some cytotoxicity assays where we're looking at dose-dependent cytotoxicity on HER2 presenting cells. We're comparing our molecule with trastuzumab, the actual drug. And you can see in 3 HER2 positive cell lines, they're about the same. The A431 cells are a breast tumor that does not have high levels of HER2, and so neither of the molecules has an effect there. We also did a PDX model. So we put tumors in mice treated. Here we did a comparison with doxorubicin because we didn't have enough of the trastuzumab. But my point is, over 30 days, the green vehicle mice clearly show a growth of the tumors. Dox shows a rest and some remission in blue, in red. And our megamolecules arrest growth and maybe show a little remission in blue. The nice thing is, as many of you know, the Dox is not well tolerated, and most of those mice don't make it through the 30-day treatment. Our megamolecules were really well tolerated. Okay, I'm going to leave here with one more example that we're extremely excited about and that I think points the way forward. So most people know about CAR T therapy. You take someone's T cells, you isolate them, you engineer them with a chimeric receptor so that the T cells have a receptor that binds to an antigen of interest. You then expand those T cells, transfuse them back into the patient, and these are now primed to recognize the antigen presenting cell, the tumor cell. Well, that's turning out to be an extremely important therapy, but probably not a long-term solution because it's complex and it's biological. Our interest is creating megamolecules that have one domain that binds to the epitope that you're targeting on the tumor cell, and another domain that binds to a T cell marker, in this case CD3. And so, as you can see on the right, can such a molecule simultaneously engage the antigen presenting surface and the T cell surface? Well, we've built these molecules, we being Shreyas, a really talented grad student in the lab. What you're looking at here in the left, we've used cell tracker dyes to label these HER2 presenting 474 cells. We've also labeled Jurkat T cells with a green tracker dye. So we mix them, wash, the T cells mostly get washed away, and you see the red cells, which are the BT474s. Now, if we do that same experiment with the megamolecule, which is intended to bring the T cell to the HER2 presenting cells, you see what we see on the right after washing. These BT474s are decorated with large numbers of T cells. So our megamolecules are recruiting the T cells in the same way that CAR T therapy is intended to operate, but with a single molecule, a single entity. It's defined, it can be delivered. You don't need to harvest cells and go through all the steps. Now, this is the first example, and it's just at the level of binding. I'm not showing T cell activity yet, but I'm more excited right now to actually increase the valency. So instead of having one Fab for CD3 and one Fab for the epitope HER2, what if we create molecules where we have multiple Fabs against the T cell surface and multiple Fabs against the antigen presenting cell? We should have a much more efficient delivery. And we're building these structures. And in fact, you can imagine all kinds of antibody mimics where you have defined scaffolds decorated with Fabs. You can imagine installing drug conjugates, but in ways that are much more controllable than what's currently done on antibodies where you get mixtures. You can imagine installing not radiolabel, but other labels for diagnostic applications. And again, the modularity and the flexibility, these are like Legos, just give a lot of flexibility in how to do that. So with that, Vadim, I think it's 10 minutes and I'm going to stop sharing and we can take a few questions, I think.
Right, let's thank you so much. This is really phenomenal. So question, so nobody has typed anything in the chat box yet. So maybe I'll just open it up to the audience. And if you have any question, just go ahead and say it.
Hi, Milan.
Oh. Hi, Sam.
Hi. This is really interesting work. Very, very nice.
Thanks.
I guess my question would be, as you start increasing the valency, then I would imagine that there is folding of the folded proteins. I mean, a double folding, right? Because you have other interactions. I mean, in the simple one, I can see it. But let's say once you go to valencies of 4, 6, 8, whatever, wouldn't you get some folded structures that then will operate very differently? And I guess along the same lines, I'm curious about if the issue of different proteins in the mega molecule having different half-lives,
Yeah.
so then the function is changing. But I mean, and in spite of all these, I think it's really an interesting approach,
Those are good questions. Thanks. So one thing that we're excited about is that we think as we increase valency and use different scaffolds, linear, cyclic, branched, there are going to be unpredictable differences both in how efficiently they serve as the link between the two cell types, but then also when we're starting to look at cell killing assays by the T cells, how effective those different links are to allow the T cell to clear the antigen presenting cell. And so the strategy is to create libraries of well-defined structures, right, and be able to see which ones are optimal in those different ways. And that's unlike an IgG where you have one shot on goal and you get what you get. Oftentimes it's good, but it's not at all clear that it's optimized. Half-lives and things, the biggest challenge we have right now is that we're building these structures out of proteins that are from bacterial and fungal sources, which means they're going to be highly immunogenic, and so they won't be candidates for human use. We've been building connectors, enzymes, and irreversible inhibitors now from human enzymes, and we think that building these structures out of human enzymes and backbones will do a lot to humanize the structures, and then we may need to use some of the other modifications and glycosylation that others have shown can be important. So a lot still to see, and I think we're going to get a lot of surprises.
Yeah. Thank you.
Thanks. Tabu, were you next?
Oh, okay. Yeah, Milan, I think the potential is limitless. The question I have, at least from an enzymatic point of view, how processive are these mega molecules in the sense that you bring these cells when the cancer cells die? Can these mega molecules bring now potential T cells or other effector cells to a different cancer cell, thereby making them more processive than just self-destruct or whatever?
Yeah, I think there are probably ways of making them multifunctional or reusable. I mean, I think one thing that we've thought about, haven't done anything, and it's related to Sam's question about half-lives. We could build into our linkers cleavable bonds, and so you could tune the half-life of the structure. There are cases, for example, in imaging where you might want to do that. If you've got an alpha emitter in your imaging, once you've localized the imaging agent and gotten an image, you actually don't want it hanging around longer. You'd rather it clear. And so you might be able to build structures that are programmed with a labile bond to start to fragment and then lead to more rapid clearance. So I think thinking about those ideas where you can play with that, but you have the flexibility because you're not really playing with the protein stability per se, but the linker integrity probably gives some general ways of optimizing on that score. Marcus?
Milan, great potential, wonderful work. Question, so maybe I missed it, maybe you mentioned it. So the building blocks, the protein domains themselves, are they fully synthetic as well, or are you just linking recombinant proteins?
These are recombinant, and so we're just most of these right now making in bacterial cultures. You could make them in other cultures, but the fusion proteins we're using, they're straight recombinant fusions.
Okay, cool. Yeah, makes sense. Thanks.
Thanks.
Thank you. So we have one more question from Jennifer Wu, but maybe we'll, and that's a good question to ask the entire panel maybe. So we'll just maybe wait for that if you don't mind.
Sure.
So I think it's a good time to go ahead with the second talk. So Mike Jewett will talk about accelerating protein therapeutic discovery and development with cell-free systems, another very exciting and quite unique approach.
Awesome. Thanks, Vadim. And really grateful for you and the Cancer Center to put this together. I think, as Milan was mentioning, as the Center for Synthetic Biology has grown, we're up to about 14 core members now, several folks housed in the medical school. We have an ongoing search with cellular and developmental biology. We're really looking to grow and make intersections and connectivities. So would really welcome partnerships and discussions on the back end. So again, thanks for organizing this. And hopefully this is really just the start to new collaborations. In my brief time today, I want to talk about two main stories in how we're accelerating protein therapeutic discovery and development with cell-free systems. And I'll describe cell-free systems and then get into the first effort, which is more of a biomanufacturing focus, where we're developing on-demand manufacturing paradigms for synthesizing therapeutics and vaccines, where you need it, when you need it. So trying to think about localized production. Then I'll pivot a little bit. And rather than focusing on what cell-free systems can make, we'll actually show how they might be able to accelerate design. And I'm going to focus on some antibody production related to COVID-19 antivirals. So just, something we all know, and I don't need to spend a lot of time on, but protein medicines have really revolutionized our ability to prevent and treat human disease from antibodies, vaccines, hormones, et cetera. And, as we motivate this first vignette, one of the challenges is that the World Health Organization estimates that about 30% of the world's population still lacks access to these essential medicines. So why is that? One of the things that we can begin to appreciate is that our current manufacturing paradigm really limits the ability to distribute protein medicines, in part because currently we make medicines in large centralized facilities, such as this manufacturing plants such as Genentech and Vacaville. They cost hundreds of millions of dollars and more. We make these protein biologics in large tanks the size of Olympic swimming pools, and then we distribute them to the point of need, typically through cold chain. Certainly a point of discussion in the recent differences between the Pfizer and Moderna COVID-19 vaccines. And so really, when you think about our ability to produce and deliver medicines, it can be limited by the expense and time required to build new facilities, as well as the feasibility of these refrigerated supply chains. Now, one way to potentially address this is using something that I'm going to call cell-free systems. So cell-free biology is the activation of complex biological processes without living intact cells. What we do is we take a living organism. Oftentimes we use E. coli, but we use many organisms in my lab. We rip off the cell wall and we collect the inside. So we collect the molecular machinery of the cell. And then we feed that the substrates, salts, DNA template necessary to manufacture a therapeutic. You can kind of think of this like taking your car, lifting the hood up, pulling the engine out, and repurposing the engine. We're basically repurposing the engine, the molecular machinery of the cell, to make therapeutics without living intact cells. And the ability to readily store, distribute, and activate freeze-dried cell-free systems by simply adding water has opened some exciting new opportunities for on-demand biomanufacturing and point-of-care diagnostics. You can kind of think of this like freeze-dried yeast you might buy from your supermarket if you want to bake bread, or if you're into brewing your own beer, you might buy from a local manufacturing shop. We just freeze-dry our cell-free systems and can make products. So we do this across a variety of medicines, both protein therapeutics, including some antibodies that have potential relevance to cancer. The quick story I want to show today is just thinking about vaccine expression. Everyone's thinking about vaccines at the moment with COVID-19. And in some, oops, I moved my slide forward. In some recent efforts with Matt DeLisa, who's at Cornell University and is a collaborator with Milan Mrksich and myself on a DITRA grant, along with a terrific graduate student, Jessica Stark, and Matt's student, Tommy, we decided we'd try to address this challenge of on-demand manufacturing. So address this issue of localized production of medicines where you need it, when you need it. The idea would be we take a cell-free system that has been enriched with glycosylation components, we freeze dry it, would allow that to be transported where you need it without refrigeration, just add water and make a medicine. If you wanted this platform to exist, you'd want it to be fast so that protein synthesis and antigen conjugation could occur together. So in this case, we're going to focus on conjugate vaccines. So the idea is you have an FDA approved or licensed carrier protein, typically one that your immune system already recognizes, that's covalently attached to an O antigen of a pathogenic bacteria, typically these sugars that exist on the outer surface of the bacteria. And so basically it's like a fingerprint. And so by giving a human this conjugate vaccine, you gain and learn and build an immunity to the O antigen so that when you recognize the pathogenic bacteria, you can fight that off. You could similarly imagine cancer vaccines or other approaches using this kind of technology. So the idea would be that it's fast. We can make the medicines in just a couple of hours, modular for the production of a variety of molecules, portable for cold chain independent distribution, safe for injection into humans or animals with minimal purification steps, and effective. And the short story is just shown here. We developed this freeze-dried cell-free protein synthesis system that could make vaccines for less than an hour for less than $1. Okay. And we, in this case, developed a vaccine, again, Extracellular tularensis is a pathogenic bacteria. And in an animal challenge model where we basically gave this mouse the vaccine over 3 doses, then challenged it with a lethal dose of the pathogen, we were able to get 100% survival, so complete protection with the vaccine that we produced in our system. So I'm just trying to share that I think we've started to set the stage for this idea of a kind of portable on-demand biomanufacturing setup for therapeutics and vaccines without a cold chain. Why that might be interesting is as you think about potential applications in your own research where manufacturing medicines, protein biologics, even thinking of some of the megamolecule designs beyond the IgG that Milan was just mentioning, we would certainly be interested in trying to think about how you might make these in a feasible way that could allow for broad access. Just to put a quick slide up here, these cell-free systems have really been able to synthesize a wide array of glycans. So in fact, we've made over 50 therapeutically relevant glycans, from bientenary glycans to minimal sialylated glycans. And so I think that there could be a lot of interest crossing a variety of medical applications, from certainly cancer to cancer vaccines and also allergy. Separately, one of the advantages of these cell-free systems is you reduce the manufacturing process to liquid handling. And so it really sets the stage for accelerating time to market. And so I want to talk about, in the second story here, just a way in which these cell-free systems are being used to accelerate time to market for medicines. Certainly, COVID-19 has highlighted the need for rapid therapeutic antibody discovery pipelines, where we obviously know that SARS-CoV-2 is decorated by this trimeric spike that's being recognized in our human cells by the ACE2 receptor. And we'd like to create neutralizing antibodies or neutralizing antivirals that could block this interaction. Now, one of the challenges is that the expression of antibodies can be a major bottleneck in the discovery pipeline, often taking weeks to months. And in fact, when you look at just DNA to a clinical dose of a molecule, this can oftentimes take 18 months in total process because of cloning, cell culture development timelines, et cetera. These cell-free systems can be faster. So even on the short term, just to manufacture medicines, rather than days and weeks, we're looking at hours. And in fact, one of the companies I've started called Swift-Scale Biologics is focused on condensing that timeline of 18 months I just mentioned down to 2 months using this technology. So we really do see it as this kind of quick approach to get there. So what we wanted to show is that we could actually express a library of antibodies that could be used to block the spike protein interaction with ACE2. And so what we did is we basically fully automated and pulled together a workflow that could be done in 384 well plates, from DNA assembly of antibodies, neutralizing antivirals, to the antibody expression, to ultimately the antibody evaluation. And I'm going to move through this kind of quickly, but just to kind of get you a snapshot of what we're doing. So the first is we have this cloning-free DNA assembly workflow that enables rapid high throughput expression of proteins. So we go ahead and acquire double-stranded DNA from a vendor, assemble that using Golden Gate assembly, amplify the template, and we basically have expressed protein within 6 hours. We've demonstrated this successfully with 400 unique different nanobodies, antibodies, scFvs, et cetera, in a reasonably short period of time. Just to show an example of what we've accomplished, here's some antibody synthesis where these cell-free systems can be used to make full-length antibody. It's a little bit difficult to see here, but we have without DTT and with DTT, we should break the antibody apart. Without DTT, we actually get some antibody. We get some of the smaller intermediate parts as well, but this major antibody band is what we were looking for. We then went on and connected this to a very rapid assay that could be used in a plate well format without being disrupted by our cell-free extracts, where what we're going to look for is binding of an antibody to the spike protein using this alpha assay that connects kind of light emission through a donor and an acceptor bead. And just to kind of show you what that does is it basically is kind of an in-solution ELISA assay, and it is designed for high throughput workflows. What we're able to show is that this alpha assay could actually quickly adapted to evaluate COVID-19 neutralizing antibodies. And in this case, what you're going to see is as you get a blocking interaction, we're going to decrease the signal. And we can, I'm showing here 5 different antibodies we expressed in our system, being able to get very nice binding affinities, fraction inhibition. But we actually scaled this to working in 384 well plates. So I'm not showing that data here. You could also actually look at a competition experiment where we're not just actually binding between the spike RBD and the antibody, but actually competing with the ACE2 receptor. We actually find that our results, the best antibodies that come out of our results, are also the ones that have the best neutralization data that's been reported in the literature. So all in all, what I hope to show you today is just that these cell-free systems are kind of transforming antibody discovery and development. We can rapidly assemble, express, and evaluate many types of binders, antibodies, therapeutics, glycoproteins. I'm re-highlighting some of these variant glycans that we've shown. And I think what we're really looking for are opportunities and connections and thinking about glycoprotein therapeutic production as well as vaccine synthesis. So very grateful, of course, for all the students, the environment here at Northwestern, and hope you will engage with us in this conversation beyond today. So thanks so much.
Thank you, Mike. Thank you so much. Really, really exciting. And I think we all can appreciate the potential, especially given what we learned during COVID. Questions? So maybe in the interest of time, we move on to the third and last talk, and then we'll have more questions.
Perfect. Yeah, hit the discussion. Sounds great.
Right. During the panel discussion. So Joshua Leonard will talk about enabling design-driven medicine with engineered cell therapies.
Great, thanks so much. I would just echo the thanks from the Milan and Mike to Vadim and team for putting this together today. So my lab is generally interested in mammalian synthetic biology, and I decided to pick a vignette today that hopefully will hit on 2 areas of potential collaborative interest in this group, involving both application to cancer therapy, which is where I'll spend most of our time, but also building technologies that could be useful for fundamental cancer research. And I'm hoping to touch on each of those possibilities here today. So within all of synthetic biology, a general goal is to make biology engineerable. And I'm going to talk about how far we've come to pushing that paradigm when thinking about engineering design-driven cellular functions here, so that we can get to the point, or how close can we get to the point, where much like an electronics toolkit, you can put things together and they work as expected when engineering something as complicated as this example of a neutrophil chasing around a bacteria, right? So I'll talk a bit about what we've invented and sort of the state of the art today, and as it applies to cancer therapy. One of the areas of interest for us right now is engineered cell therapies for a lot of applications. And an exciting part of this field is that these are no longer theoretical. These are in the clinic and increasingly in commercial translation as well for a variety of applications, but probably none more so and more relevant today than the use of engineered cell therapies to treat cancer. So everyone in this audience, I presume, is aware of the promise of the first big winner in this space, CAR T-cell therapies. And Milan gave a nice example of how T-cell therapies in general can be used to recognize specific targets. I'm using this, and of course, this is already in a good amount of commercial translation, dozens of companies now looking at different ways, probably hundreds of trials in just this kind of space alone. What I want to point out is that that's still a relatively small fraction of the cancer market, in part because these work really well for certain types of cancers. And right now it's hard to get these to work, for example, for solid cancers, although a lot of progress is being made there. The other big window is that every one of these therapies relies upon foreknowledge of that molecular handshake that must occur between the engineered cell therapy and the antigen or combination of antigens on the tumor surface. And thankfully that works for a lot of cancers, but there are many cancers that lack a unique surface antigen and others for which that antigen goes away over the course of therapy or remission in response to an antigen-specific treatment. And so the area we've been working for a while on is trying to figure out how to make cell therapies that are smart enough to recognize a tumor, but not through the molecular handshake. And that's really what's inspired the suite of technologies I'll talk about today. So for example, we've known for about 2 decades now that if you were able to be very small and get to every single tumor and inject immune potentiating factors at the tumor site, there are tumor reactive T cells in many cancers and other cells that if you were to deliver enough stimuli locally, you can engender a remission, you can engender a response by those existing cells. The problem is we just don't have the ability to do that in a way that works for metastatic disease, disseminated cancers. And in particular, we don't have the ability to do this in a way that balances safety and efficacy. And so that was the motivating statement for us, was that we know this is a therapeutic modality that could work. And so could we use engineered cells as the vehicles to find those disseminated tumors and administer potentiating factors that would work locally, but that can't be done in a distributed administration fashion. So when thinking about a complex problem like that, you need to break it down like any engineering problem into a series of manageable steps. And for us, that means thinking about sensing, processing, and actuation as a way of composing the kind of design statement like we had on the last slide here. When we started working on this, we really lacked all of these things for the most part. But we started off with trying to solve the problem of building engineered sensors that, for example, could stand in for native receptors, which just oftentimes aren't sufficiently facile to be incorporated into a complex biological function here. So the first technology that we invented a couple of years ago is called the MESA, the Modular Extrasensor Architecture. It's a fairly similar, simple concept in that there are 2 engineered receptor domains where upon ligand binding, and this is an extracellular protein ligand, like a cytokine in this case, you get dimerization of the receptor domains, bringing one into proximity with the other. You have an intracellular cleavage event, and that releases, in this example, a sequestered transcription factor. And so the downside of a synthetic technology like this, or kind of a Frankenstein synthetic technology like this, is that it's much harder to get it to work compared to just co-opting native receptors. However, we spent a lot of time getting that part solved. And then you get to benefit from the modularity in a system like this, which is that once you understand what's happening here, you can swap out the domains and still have a functioning receptor in a way that wouldn't be possible with, say, fusion proteins based upon native receptors. And so I'll show you just a couple stories here. The first one we showed was that you could take that core concept and apply it to something of use for immunotherapy. In this case, we showed we could engineer cells to sense VEGF, which is elevated in the blood, in the tumor microenvironment. Even in patients who have solid tumors, you find more VEGF at the tumor site than in the rest of the plasma. And we showed that we could then couple that sensing to the release of, in this case, a Cas9-based transcription factor, which turned on the endogenous IL-2 locus. So first thing, that's a novel input-output function. There's nothing that senses VEGF and then makes IL-2. So this speaks to the idea of, wouldn't it be interesting just to find out what happens if a cell were to do that? It's of course useful also in the sense that sensing a tumor environmental factor and producing a T cell stimulation cue would be translationally relevant. And I guess importantly in this story, we showed that once you figure out how to get those parts to work, if you then pop it into different cell types, you don't have to reinvent the wheel every time. So here we showed that putting that same machinery into an engineered cell line, you can confer this IO capability to human AGK293 cells. And when you pop those over into Jurkat T cells, they also can exhibit this input-output relationship here. So that's the idea here, is that there's a context generalizability with these modular parts that has a lot of advantages for thinking about applications to new spaces. So now MESA is just one of a handful now, and really a growing list of synthetic receptors you can use depending on what kind of functionalities you want to put together. CARs are, of course, the ones we mentioned. SynNotch is a technology invented by Wendell Lim's lab that senses, in a similar way to MESA, surface-bound ligands rather than the soluble ligands that we can sense. But we now kind of have a toolkit for putting together these different sensing functions here. I'm not showing a couple stories, but one of the cool things we've shown in a couple of recent stories here is that it really drives home the idea that going from proof of principle to translation faces many challenges. One of which is that a lot of times with engineered functions that are based upon natural receptors, if they work as desired, you're great. And if they don't work as desired, you're stuck. And what we showed in a series of recent stories is that by contrast, synthetic receptors give you the ability to optimize specific performance characteristics in a way that lets you actually meet the needs of a given application here. And so I'm just using a cartoon to kind of show you how you can map that anatomy with a biophysical characteristic and a performance goal that is needed for an application. So the last thing I'll show you is focusing on this next step of processing information here. And so again, when we started this, there wasn't really a toolkit here. So we built one based upon the very commonly used in biology zinc finger motif. And so the first thing we did was build a giant library of these, characterize them quantitatively. That immediately solves one goal. Now we have parts and you can pick and choose. And if one wants to, they can use our characterizations design stuff. But I think the more exciting part of this, and what's much more of an engineering side of this, is that we took a few extra steps. One of which is that we built computational models which help you understand why each of the several hundred things in the library works the way it does, and works differently, and to some extent the biophysical mechanisms that each one of these things uses. That's what gets you to the point where you have parts that, for example, we share with the research community via AdGene. And the last arc here is a recent story where we showed, we tested the proposition, are we really doing engineering? Can we not just explain what's happening, but start with the design and then build it and see if it actually works? And so in a story that will hopefully be out in Science Advances in a couple of weeks here, we showed that we, for the first time, can do this. And in fact, it's the first time in my career, you build something and it works the very first time, exactly basically the way we expected. And in this case, the thing we're trying to build are complex logical functions, which is really the biological analogy of a sophisticated function. If you want your cell therapy to become activated when it sees this antigen, but not if it sees that antigen, or it can see either of these, but not either of these, that's the kind of logical statement that inspires this kind of genetic circuit design. And we showed in this case that we can make designs that work something like half a log or even up to a log better than the previously most complex things you can do, which means we now have a hammer that we can use for a lot of different projects. And we have some in mind, but would love to collaborate on thinking on some other ones. So I'll just end that the data I'm not showing are really the most cancer specific one, in a sense that we have an ongoing project with Bin Zhang, who's in hem-onc and microbiology immunology, with engineering, in this case, natural killer cells to treat cancer using this microenvironmental sensing approach. One reason we're interested in that beyond the one I mentioned is that these are candidate off-the-shelf products so that they're not patient-specific. They can potentially bring down the cost of these manufactured cell products by an order of magnitude or more. We're also using these as part of some other collaborations to study some some fundamental questions in cancer biology, but really that's an untapped part of the potential space here that I'd love to use this discussion to kind of stimulate. If you have a question, if you have an monitoring of a complex function inside of an animal that you think you'd like to see, but you can't see, that's where a lot of our tools are being used right now. And we have some related work looking at other types of biological therapeutics, such as engineered exosomes, which are nanoscale vesicles that we're using as both functional mediators, outputs of cell therapies, and potential windows into what's happening biologically in complex organism scale environments. So I'll end there and say that if anybody's interested, I went to a meeting about a month ago at the NIH focusing on the interface of cancer and systems and synthetic biology. So be happy to share those materials with anyone interested. And I'll end by just, again, thanking my team. And I'll open the floor to any questions folks might have. Thanks.
Thank you, George, so much. Any questions for George? right. So maybe that's a good time to open up a panel discussion. So this is the time when everybody should feel free to ask questions to any of our three synthetic biology experts here. And maybe I will kick it off with a good question that Jennifer Wu posted on the chat in the chat room. And her question is, it's fantastic talks. What is the regulatory pathway here? So if anybody wants to tackle that.
I guess there are two sides, actually three sides, each of us. I can say for megamolecules, you know, one of the benefits of these structures is that they are discrete structures. So it's not a mixture of structures like you have with the antibody drug conjugates that are now being used and in trial, but these are discrete structures. So I think they would go through the same process that, you know, all biologics go through. In a pretty similar process. I think for us, the real challenge because we're not ready for that yet, is what I've referred to earlier in terms of the immunogenicity. I think we're going to have some good building blocks that address and minimize problems that we would definitely have with our current ones. But in terms of approval, I think they'll be treated just like all biologics.
I can jump in. And so for the cell-based therapies that we're working on specifically, these have already in concept been used in a less, let's say, sophisticated form in human trials. So the base translatability to humans has been shown. So we have safety and efficacy data, and especially safety is important, I think, in that context there. There are also commercial partners who are using, or commercial entities that are using both primary and immortalized natural killer cell products, and they're pioneering some of those regulatory questions here. So they haven't reached regulatory approval yet. That's one answer to that, but we're starting to get an increasing pile of human data, and safety doesn't seem to be the issue. The question of immunogenicity is also an issue with cell products. So there are some designs we're doing where there is nothing that should be non-native. We use genetic engineering approaches, but we can use parts that are native or that are nucleic acid based to sort of circumvent that question. The other angle is that our translational model for these NK cell therapies is really premised on the idea that if immunogenicity is an issue, that doesn't preclude efficacy, because effectively our cell products, and let's compare those to a car product, they're meant to be used transiently. They stick around for a couple of days. You may have to administer them more than one time, although it's not clear that's necessary. So rejection of the cell product and really vector-based immunity may be beneficial in the case of treating cancer specifically. So we're kind of hedging our bets. We as a field, not just my lab, are hedging our bets on that particular one there. But we think that like this kind of transient cell products in general has some advantages in the sense that you don't necessarily have to stick around and avoid immunity unless you have to do repeat administrations.
Okay.
I can comment on the work I prepared. I think from the point of view of just using cell-free systems to accelerate design of, let's say, cancer-based antibodies, ultimately the antibodies would probably be manufactured as they are in CHO cells or others. So I think the regulatory path there is pretty straightforward as the discovery tool. On the manufacturing side, cell-free systems are being used for the first time now to manufacture products. In fact, the strains we use have been engineered to have significantly reduced endotoxin levels, et cetera. And in fact, the very first cell-free manufactured antibody drug conjugates are going through the FDA right now by a company called Sutra Biopharma. So I think once that path has been paved, there is actually the potential also to imagine these cell-free types of approaches for manufacturing on demand.
That makes sense. Thank you. So I had another question. So what do you see as the next, I guess, challenge, roadblock, bottleneck, or a limiting piece, or a missing piece? Is it in implementation of synthetic biology to cancer? Is it identifying critical questions that need to be addressed? Actually developing, let's say, therapeutic agents, so more translational work? Is it transition to human studies? This is just a few potential examples. What I guess, what would you see as the most challenging next step for you?
Mike, why don't you take it first?
Yeah, I think, you know, when I think of synthetic biology as a field, really, it's really grown out of this ability to read, write, and edit DNA at capacities and throughputs that we haven't had before. And because of that, it's brought in this new wave of, I think, engineering design to thinking about therapeutic problems, addressing cancer, et cetera. I think as a result of that, one of the areas that I think is a real opportunity that's going to emerge is at the intersection of data science and AI and machine learning, informing large scale either syntheses approaches of biologics, or in the very beautiful work that Josh highlighted that worked on the first time as we start thinking about this, how do you get designs to actually work more frequently when we want them? And so I think one area is going to be this combination of the right problem with the tools of synthetic biology being accelerated by data science and AI and machine learning. The other side, I would say, is still that translation component. So so that you mentioned as your last point. So I think for me, those are two key areas. But how do we get from that discovery to translating it to those early models, I think is still a challenge.
Hmm.
Maybe I could build on that. I think at this meeting, for example, in similar ones, one thing that comes up is that, as engineers, we're oftentimes focused on creation of new capabilities, but practitioners have a problem with the number of tools that exist already, in the sense that if you have radiotherapy and chemotherapy and checkpoint inhibitors, that's already a lot of moving parts. People are nodding their heads. And so this I think, is an area where what Mike mentioned could be extended to the clinical feedback loop also, in the sense that I think knowing how to identify where the gaps are and focus the engineer's attention on the places where there is an unmet need, that's a real big opportunity that could be extended to the use of clinical data, feeding it back to the bench scale also.
Yeah, and I think the three sketches today, in my mind, illustrate the three ways that SynBio offers some new ways forward. I think one is that in our case, we're able to go beyond what biology is programmed to do with antibodies, making antibodies that are multivalent, multi-specific, derivatized or functionalized with other things in ways that are still extremely challenging to do if you're starting with an IgG. And so it's taking biology and it's kind of adding more options for how you engineer biology. In Mike's case, it's really about, the manufacturing side. Once you have something that you want to distribute, if you can manufacture locally, if you can rapidly establish a new manufacturing capability, because this is a really flexible approach, there's all kinds of benefits, and these mRNA vaccines are showing us, if you're in a city, you're going to have access to it. They're going to be lots of part of our population and the global population that are going to have a hard time getting these things because they don't have the infrastructure for the cold chain. And then with Josh, can you mimic the immune system with new engineered cells so that these cells turn on their activities when it's needed and where it's needed. There's a feedback that kind of knows when to turn off their activities too. And those are just novel ways of thinking about therapeutic intervention. And so I think all three of those are, represent what's new, represent new approaches and directions that have grown out of SynBio.
Okay. Well, thank you. So there's one other question is from Debo. So Debo is wondering whether the cell-free system will be able to produce large-scale biologics for therapeutic use. And he was wondering about the superiority or advantages of cell-free systems over purified large-scale systems as well.
Yeah, thanks. Great question. In the context of cell-free systems, I think the long-standing belief is, these are great for 10 microliter reactions for discovery. We discovered the genetic code in crude extract systems, and that works great. Over the last 2 decades, actually, a very interesting technical renaissance has enabled large-scale manufacturing using cell-free systems. So on the order of 100 to 1,000 liter reactors. So we're not to 10,000 liter reactors, but we're into the 1,000 liter reactor scale. And I think that could grow. So imagine, for those of you who, we have a crude extract system. It's like running a PCR reaction cost effectively at the 1,000 liter scale. So we're leveraging things like natural endogenous entropy metabolism and other components. And so I do think, it's not going to replace CHO cell manufacturing of antibodies, and that's not the goal. I think it's going to allow us to produce new therapeutic modalities more efficiently, maybe look at things that need an expanded chemistry, or perhaps homogeneous glycan production is something we're looking at. In terms of superior over purified system, in the end, you would be delivering a purified therapeutic or biologic. So we do purify the molecules. So I might not have been clear about that. So if that's what the question was, we have, for example, purified our vaccines before injecting them into mice, as just one example. So we are going to have to purify the proteins, I think, and that would be expected. But it is interesting to see how far the field has come.
Thank you. Josh, Milan, you think you wanted to add to that?
I covered it.
Thank you. So we are approaching the top of the hour. I wanted to make sure that everybody who had a question has a chance to ask it. Is there any last minute burning question anybody wants to ask? Well, I want to thank, on behalf of the Cancer Center, on behalf of everybody who is here, Mike, Josh, Milan, thank you so much. This is really extremely exciting I think for all of us to watch in real time how synthetic biology at Northwestern is emerging as the world leader in the field, and hopefully in cancer research of the future. There's a clearly great potential. Thank you so much for giving fantastic talks, and thank you for being at Northwestern. And I would like to encourage everybody who is here, who listened to these talks, if you have any follow-up questions, if you have any follow-up unmet needs that you would like to discuss with our synthetic biology group, if you would like to work on translation or discovery, please feel free to get in touch with Milan, Josh, or Mike. You're welcome to get in touch with me, and I can coordinate the further communications as well. Let's try to make it great. I think it's going to be the beginning of a beautiful friendship. Thank you, everybody.
Thank you, Vadim.
Thanks, Vadim.
Thanks all.
Bye, everybody.